Hello. Gene transfer is the method of transferring DNA into a cell. We can make the recombinant DNA by combining two different DNA from different organisms. In a typical way, what we do, we will take the plasmid DNA or the vector DNA from the bacteria and we will insert the foreign DNA into the cell. We can use the restriction nuclease to cut the DNA and we can use the ligase enzyme to ligate the DNA. Once the restriction and the cutting and the ligation is over, we will get a recombinant DNA which is containing both the parts from the bacteria and it also containing the foreign DNA from different organisms. So this recombinant DNA need to be transferred into a cell. The process of transferring recombinant DNA into a cell is called gene transfer. In this session, I am going to give an introduction about gene transfer. So gene transfer is the introduction of DNA into a cell. So we are transferring the DNA from this test tube into a cell. So it's insertion of unrelated DNA because we have a recombinant DNA. The recombinant DNA itself containing DNA sequences from two different organisms. So it's the DNA composition of the recombinant DNA is different from that of the host DNA. So we are inserting an unrelated DNA into the cell. So that's the process of gene transfer and it's an example of horizontal gene transfer. We know that the gene transfer can be of two different types, vertical gene transfer and horizontal gene transfer. The vertical gene transfer is from parents to offsprings generation after generation. So that's what we call the vertical gene transfer. So from the parental generation to F1, F1 to F2, F2 to F3. So that's the vertical gene transfer. The horizontal gene transfer occurs the transfer of DNA from two different organisms which is not related in a familial tree or they are two parallel living organisms. That's why it's called horizontal gene transfer. So the horizontal gene transfer can be done between two different class of organisms like the transferring the human DNA into bacteria or transferring the bacterial DNA into a plant. So both these organisms not related in a family or they are not a part of the same species, still we are able to transfer the DNA from one organism to the another organism. So that type of process is called horizontal gene transfer. So the gene transfer what we are discussing here is actually the gene transfer and the DNA or the gene we are transferring from one organism to another is called transgene. So gene which is transferred is called transgene. So in the horizontal gene transfer, the transgenes are transferred from between organisms in a manner other than the traditional reproduction. So traditional reproduction is the vertical gene transfer. The gene transfer we are actually talking here is the horizontal gene transfer, transferring DNA from one organism to another. The it's a the process of genetic uptake is referred to as transformation. We already know the process of transformation, the way organisms take up the external DNA or the process of gene transfer is called transformation. The organism said to be transformed after taking up the DNA or transferring the DNA. So we have the transformed cells or transformed bacteria. The process is called transformation in case of gene transfer. So this is the diagrammatic representation. It's a typical diagram. We are taking the plasmid DNA or the vector DNA from the bacteria and we are combining it with the insulin gene from the human cell. So the insulin gene is isolated and inserted into the plasmid. So after insertion, we get a recombinant plasmid or recombinant DNA because it is having components from the bacteria as well as from the human being. So this construct recombinant plasmid is then inserted into a bacteria. This process of insertion is called transformation or gene transfer. So how can we insert a DNA into a cell? DNA will be like a naked DNA. It is not associated with any protein. It will be in a test tube. We need to insert the DNA into a cell. Once it is inside cell, it can function. It can make a protein, it can make an RNA, it can cause other changes in the organism. So in order to do that, we need to have some techniques to insert the DNA into a cell. So what is the purpose of transformation? The transformation can be used for cloning, so increasing the number of DNA or the copies of DNA. We can use a transformation, or we can use it for the production of large amount of proteins, especially for the recombinant DNA protein. In the previous example, we have seen the production of insulin. Once the insulin gene from the human being is transferred to the bacteria, we can actually produce the insulin for various therapies, for diabetes or something like that by culturing the bacteria. So we can use it for culturing, producing large amount of recombinant protein. We can study the gene function and regulatory aspects in the bacteria. Once we have cloned it, then we can study the characteristics of gene promoter, the ability of promoter. Then it can be used for gene therapy. Then it can be used for crop improvement like herbicide resistance, pesticide resistance, weed resistance, etc. can be made. Then it can be used to introduce novel traits to an organism. And in the last chapter, we will be discussing the applications of recombinant DNA technology. So all of the applications of recombinant DNA technology is due to the transfer of DNA from the test tube into an organism. So it has various applications, and we will be discussing it in the last chapter. So the transformation, the process of gene transfer involved two stages. One is transfection stage and second is integration stage. Transfection is actually the introduction of DNA into the cell. The DNA which was in the test tube, after transfection, it is inside the cell. So that is called the transfection stage. In the second stage, integration stage, what happens is like the DNA inside the cell get integrated into the chromosomal DNA of the organism or the bacteria. So that is called the integration. So in the gene transfer, the first step, the transfection stage is essential, but integration stage is optional. Some of the DNA do not need integration. It can function as an extra chromosomal DNA. It can replicate by itself. But some for other vectors, there won't be any origin of replication within the vector. So we need the DNA to be integrated into the genomic DNA. So these are the two stages, transfection stage and integration stage. And the transfection is much more efficient than the integration. So integration is a rare event. For the integration, there should be a homologous recombination between the foreign DNA and the host DNA, and its frequency is also less. But transfection we can achieve by using different techniques. The transformation leads to changes in the recipient's genotype, and it may acquire extra traits. And this transformation can be of two different types: transient transformation and stable transformation. In the transient transformation, as the name implies, it will cause only transient change or temporary change in the properties of the cell. After that period, the cells will not have any effect. The DNA, transferred DNA, do not have long-lasting effects in the cells. So the genes which is within the recombinant DNA may express, it may produce the encoded protein, and it may not have the long-lasting effect. It won't be producing a cell line or prolonged duration. The expression won't be there. So it's a rapid way of analyzing foreign gene and the gene production within a cell, but the expression is transient. In the second step, the stable transformation, the DNA get integrated into the genome and it inherited as a cell line, and we can actually multiply the cell line and all the descendants will have the property, and it results in formation of cell line. Then it used for the production of large amount of proteins for a prolonged duration of time, and the frequency of stable transformation is really low. So the transient transformation can be achieved easily, but the stable transformation is difficult. And we can actually calculate the transformation frequency from this equation. So transformation frequency is equal to number of transformants into final volume of recovery divided by microgram of plasmids used into the volume of pellet. So anyway, the transformation efficiency will be very less. So the best chance is 1:1000. If you are transforming 1000 cells, one of them will be transformed. So why the transformation is a rare event? Why the efficiency is too low? There are different reasons for a lower transformation efficiency when we are working with the bacteria, eukaryotes, or the plant cells, or whatever it may be. As the organism becomes higher, the complications, it will be more complicated to achieve the gene transfer. Why it is so? First reason is the negative charge of DNA and the plasma membrane. The DNA is having phosphate group, ribose phosphate backbone the DNA has. So the phosphate is imparting a negative charge to the DNA, and the plasma membrane is made up of phospholipid bilayer. It also has the phosphate, so it is also negatively charged. So when the DNA and the plasma membrane come in contact with each other, they will repel because of the same charge. So that's one reason why the transformation efficiency is low. Other thing is that attaining transformation in eukaryote is way much difficult than in prokaryotes. The prokaryotes, it will have a cell wall and a cell membrane. That's it. Once it passes the cell wall and cell membrane, it is inside the cell. But in eukaryotes, we have another hurdle, that is the nucleus. So once it passed the plasma membrane, it also need to be escorted into the nucleus. So there is two barriers, and the transportation to the nucleus is highly selective. So achieving transformation in eukaryotes is difficult. One other related reason is like the bacteria are used to be very resistant to various techniques we are using for transformation. But the animal cells and the plant cells in protoplasm form is used to be very sensitive. So we cannot use the harsh technique for transforming the higher cells or higher eukaryotes. Second thing is the fate of the foreign DNA. Once the DNA came into the cells have various machineries for degrading the foreign material. It has a cytoimmune system. It will be activated and degrading the DNA. So that will determine the fate of the DNA. So it may be in bacteria or eukaryotes, it can act as a independent genome, or it can be integrated into the chromosomal DNA. But there are also some systems to degrade the DNA. So it's a tough task for the DNA. Then finally, the monitoring the transformation process. As the frequency of transformation is very low, finding the cells which is actually transformed is very difficult. We need to have the selectable markers. We need to have the markers which show different colors, so that we can actually find the transformed cells from the huge number of untransformed cells. So those are the reasons why the transformation is rare event. One is negative charge of DNA. Second is the fate of DNA is it has different fate. It can be degraded. It can be chipped out, or there are different enzymes to degrade the DNA inside the cell. And finally, the monitoring transformation is difficult. Gene transfer strategies can be divided into three. So different methods we are using can be divided into three. One is direct DNA transfer, second is transfection, and finally we have transduction. So direct DNA transfer is actually the physical methods. We have so many physical methods in which we can transfer the DNA from the test tube into the cell. Those methods are called the direct gene transfer. In transfection, we are actually inducing the cell to take up the DNA from outside. So we will be inducing the maybe we will be adding chemicals into the medium so that the cell will become more permissible, or it will we will induce the cell to take up the DNA from the outside world. Finally, we have transduction. In the transduction, we are using biological agents or biological vectors like virus or bacteria to insert our DNA or to transfer our DNA into the cell. So if any biological agents like virus or bacteria is used, it is called the transduction. So these are the major methods of or strategies of gene transfer. So here we can see the chemical methods, we have the physical methods and vector mediated biological methods. So this is a diagram. Gene transfer can be a direct method or indirect. In direct, we have physical methods and chemical methods. In indirect method, we have viral mediated gene transfer and bacteria mediated gene transfer. The chemical induced gene transfer is also called a direct gene transfer sometimes. Direct gene transfer, foreign DNA is directly introduced into the host genome. It is also called vectorless gene transfer. It doesn't mean that we don't have the cloning vector. It means that we are not using any biological agents like bacteria or virus. The physical and chemical methods are classified under direct methods, but some of the chemical methods can induce the DNA cells to take up the DNA. So that's despite of that difference, it is also called a direct method of gene transfer. Then these methods are the direct gene transfer methods are simple and effective, and it is very cost effective too. And they may lead to the rearrangements and gene silencing because of the non-specific nature of direct DNA transfer. Thank you.